Assalamualaikum and a very good morning to our lecturer, Professor Madya Dr. Khairul bin Osman and the fellow friends. Today, we are the Group 1 wants to present about the DNA is genetic material. Before we jump to this experiment, let me explain to you a little bit about the protein and the DNA. During 1930s until 1940s, most genetics paid a little attention to DNA and convinced a genetic material must be a protein. The first reason why they choose a protein as a genetic material because protein is composed of many structures, which is primary structure, secondary structure, tertiary structure, and quaternary structure. Other than that, the gene of material controls the production of the protein, so that's why they say that protein is genetic material. The second reason why protein has been chosen as a genetic material because a protein are made up of 20 different amino acids that show that the protein is more complexity and diversity compared to DNA or the other molecules. Other than that, the protein also abundant in the cell, and that's why they said the protein is genetic material because they did not see the DNA in the cell. So, why the scientist does not take DNA as a genetic material? Because DNA only composes a double helix structure, which is simple from the protein, and it has only a 4 bases, which is cytosine, guanine, adenine, and thymine. Other than that, DNA cannot be seen in the cell because it's placed in the nucleus inside the cell. So, the protein will be chosen to be a genetic material. At that time, the role of DNA were not widely noticed by almost of the geneticists. Until in 1928, one of the experiments has been done by the Frederick Griffith to show that DNA is genetic material and not protein. Frederick Griffith made a curious observation concerning 2 strains of pneumococcus bacteria. Protein is composed of many structures, which is primary structure, secondary structure, tertiary structure, and quaternary structure. Other than that, the gene of material controls the production of the protein, so that's why they say that protein is genetic material. This is the conclusion from the Griffith experiment. The lack of the Griffith experiment is he didn't know what the substance that caused the transformation to occur until at 1944. Those of the scientists Avery, McCarthy, and MacLeod identified Griffith's transforming factor is a DNA. They get this result through a series of careful experiments where they get the sample from the Griffith experiment. In their experiment, they try to lyase the smooth-strained cell and separate the cell contained into the several fractions. The fraction is lipid, protein, carbohydrate, DNA, and RNA. Each fraction were tested to see which one of these fractions will cause the transformation to occur. They test each fraction by treated the sample with the specific enzyme that can destroy RNA, protein, DNA, lipid, and carbohydrate. And the results show all the strains that contain the DNA will show the result that the culture will contain the transformed S-strain bacteria, which is they can form the smooth strain cell into that culture. And the only one of the tests cannot transform the smooth strain cell is the strain that cannot contain the DNA. And this is show that the DNA that caused the transformation to occur. This experiment is continued by the Alfred Hershey and Martha Chase in 1952. The experiment performed a series of elegant experiments on the reproduction of viruses that infect bacteria known as bacteriophage. In order of the bacteriophage to grow, it needs the host cell. So, this phage reproduce inside the bacteria cell, eventually causing the cell to break open and release large number of new viruses. So, the study shown only part of an infecting phage enter the cell, and the genetic material should be included in that portion. In this experiment, what they are trying to do is they label the viral protein of one of the phage with sulfur 35 and a viral DNA to another phage with phosphorus 32. They label with this radioactive isotope because protein contains sulfur as part of amino acid and DNA contains a phosphorus group. The phage then attached to the bacteria by agitating the sample in the blender, then centrifuge to the sample and give a pellet and the supernatant fluid. As a result, the phage that labeled the protein with sulfur 35 found radioactivity in the supernatant. This will indicate the protein had not entered the cell. On the other hand, the phage labeled DNA with phosphorus 32 found that radioactivity associated with the bacteria reaching in the pellet. This indicates that DNA has entered to the bacteria cell. So, in this experiment, Hershey and Chase conclude the phage inject their DNA into the bacteria cell, leaving most of their protein outside. So, the conclusion for our experiment is those scientists or geneticists that conclude the genetic material is the DNA and not a protein.
Now we are going to discuss how RNA is the genetic material in the tobacco mosaic virus. As we know, RNA is ribonucleic acid, and we will now refer to the tobacco mosaic virus as TMV. So, TMV was the first virus to be identified over 100 years ago. It is the first RNA virus genome to be completely sequenced, and it is a type member of a large group of viruses within the genus Tobamovirus. In the first outbreak in South America, the disease showed that it was an agent too small to be bacteria. Some surmised that it could have been an unculturable bacteria, but no evidence was found. Hosts are usually tobacco plants, tomato plants, cannabis plants, and also other solanaceous plants, such as orchids, as shown below. Infected plants will usually have a mosaic pattern on the leaves, hence the name of the virus. It will have mottling of the leaves, yellow tissues, delayed ripening of fruit, stunted growth, and also discoloration of fruit as shown on the tomato. So this virus is usually spread by an infected leaf rubbing against a healthy leaf, or contaminated tools, or possibly a worker who smokes an infected tobacco cigarette and then who handles plants. Also, it can be transferred when a contaminated seed coat germinates, hence it will be developed into a diseased plant which will infect other plants. But scientists from Germany and the US proved that only the RNA is infectious. As we can see here, the TMV virus is rod-shaped and it is highly stable. If purified, it can survive more than 50 years in dead tissue or in a lab at 4 degrees Celsius. It is also thermostable, which means that on a dried leaf, it can withstand up to 50 degrees Celsius for almost 30 minutes. It has been found that it is very stable due to the densely packed structure of the viral particles, which consists of a single genomic RNA molecule enclosed in a cylindrical protein coat. The RNA molecule is shown here in red, and the protein coat is shown in blue. So this molecule is a rod with a tight array of about 2,130 identical coat protein subunits, and each will contain about 158 amino acids. When it comes to the actual size, one rod will actually measure about 300 nanometers in length and 18 nanometers in diameter. This rod will have a hollow middle with the RNA in it. So the coat protein will cover about 6,400 nucleotides, which are made up of adenine, uracil, guanine, and cytosine. These nucleotides will encode for 4 genes, 2 of which are replicase associated, which means that they can be directly translated from the TMV RNA. The 2 others are from sgRNAs, which are subgenomic RNAs, and will code for the movement protein and the coat protein. TMV assembly, scientists have shown that it is actually a 2-step process, whereby proteins will form 2 discs of about 17 subunits each. An RNA initiation sequence will bind to the center of the ring, and this will cause the disc to dislocate, hence forming a lock washer-shaped ring of 16.3 subunits per turn. As it is shown in the diagram, it is not a complete ring, but it is rather dislocated at the end. The remaining protein subunits will stack on top of this structure until the RNA is completely covered, so it will form a helical-like structure. This process has the advantage of ensuring that only virus RNA is packaged due to the initiation sequence of the RNA at the beginning. When it comes to the TMV infection, the protein coat is very helpful in that it protects the RNA from being destroyed by enzymes. But then again, it also poses a problem, whereas it has to be removed when the virus enters a plant cell before the virus can spread. The capsid protein has several clusters of acidic amino acids, which are stable outside of cells where the calcium levels are high, but inside the cells where the calcium levels are low will repel each other. So this repulsion will loosen the first few capsid proteins, hence releasing the end of the RNA. The ribosome of the host plant will then move down the RNA strand, creating the first set of virus proteins. All the while, it will displace the remaining capsid proteins and expose the whole RNA molecule. So I should now explain that RNA actually exists in 2 forms, where it can be positive sense or negative sense. The positive sense viral RNA signifies that the viral RNA sequence may be directly translated into viral proteins, which means that the viral RNA genome is the same as the viral messenger RNA, which is to be translated by host ribosomes later. These positive sense viruses do not need RNA polymerase packaged into the virion because it can be directly processed by the ribosomes. On the other hand, negative sense RNA is complementary to viral RNA and needs RNA polymerase to form positive sense RNA before translation by host ribosomes can occur. These negative sense RNA cannot be encoded to protein directly, and they must carry RNA polymerase in virion in order to infect the plant.
At that time, the role of DNA were not widely noticed by almost of the geneticists. Until in 1928, one of the experiments has been done by the Frederick Griffith to show that DNA is genetic material and not protein. Frederick Griffith made a curious observation concerning two strains of pneumococcus bacteria.
Now we will discuss about the structure of DNA. As we know, DNA is the deoxyribose nucleic acid.
So, the conclusion for our experiment is those scientists or geneticists that conclude the genetic material is the DNA and not a protein.
The DNA nucleotide. This picture shows that the DNA strand is made up of repeating molecule called nucleotide. Each nucleotide composed of phosphate group, sugar, and nitrogenous base. Nitrogenous base. DNA has four bases which are adenine, thymine, guanine, and cytosine. All of these bases are divided into two categories, which are purine and pyrimidine. Purine is adenine and guanine, while pyrimidine is thymine and cytosine. Picture shows the structure of purine and pyrimidine. We can see the difference between purine and pyrimidine in their structure. The purine is a double cyclic compound while pyrimidine is a single cyclic compound. Chargaff rule. Chargaff rule is used in the base pairing in DNA, which state that adenine must pair with thymine, guanine must pair with cytosine. The amount in a given DNA molecule will be about the same. This picture shows the hydrogen bond between the base pair held together by hydrogen bond, 2 between A and T, and 3 hydrogen bond between G and C. Pentose sugar. DNA has a deoxyribose sugar. DNA linked by phosphodiester bond. Pentose sugar is one of the backbone of DNA. The linkage between nucleotide occur between the 5' and the 3' position on the sugar group. One end has a free 5' end and the other has a free 3' end. Nitrogenous base. DNA has four bases which are adenine, thymine, guanine, and cytosine. Picture shows the structure of purine and pyrimidine. We can see the DNA has a double helix structure which the base is complement of each other, C and G, A and T.
So now we'll discuss about Watson and Crick and the double helix DNA. Chargaff's realization that the AT and CG bases combined with some crucially important X-ray crystallography work by the English researchers Rosalind Franklin and Maurice Wilkins contributed to the Watson and Crick's derivation of the dimensional double helical model for the structure of the DNA. The double helix structure of DNA was proposed by Watson and Crick in 1953. Only using cardboard cutouts representing the individual chemical components of the four bases and the other nucleotide subunits, Watson and Crick shifted molecules around on their desktops as though putting together a puzzle. They were misled for a while, but upon the suggestion of American scientist Jerry Donahue did, Watson decided to make new cardboard cutouts of the two bases to see if perhaps a different atomic configuration would make a difference. It actually did. Not only did the complementary bases now fit together perfectly, which is A with T and C and G, but the structure also reflected Chargaff's rule.
The DNA nucleotide. This picture shows that the DNA strand is made up of repeating molecule called nucleotide.
Each nucleotide composed of phosphate group, sugar, and nitrogenous base.
They test each fraction by treated the sample with the specific enzyme that can destroy RNA, protein, DNA, lipid, and carbohydrate. And the results show all the strains that contain the DNA will show the result that the culture will contain the transformed S strain bacteria.
Picture show the structure of purine and pyrimidine. We can see the difference between purine and pyrimidine in their structure. The purine is a double cyclic compound while pyrimidine is a single cyclic compound. This picture shows that the bases are paired by hydrogen bonds and the van der Waals forces are not included in the picture. The DNA helix has uniform width of 2 nanometers and adjacent bases are 0.34 nanometers apart. Most DNA double helices are right-handed. That is, if you were to hold your right hand out with your thumb pointed up and your fingers curled around your thumb would represent the axis of the helix and your fingers would represent the sugar phosphate backbone. Only one type of DNA, which is the Z-DNA, is left-handed.
So the closure the
DNA has a double helix structure which the base is complement of each other, C and G, A and T. Okay, done.
When it comes to the TMV infection, the protein coat is very helpful in that it protects the RNA from being destroyed by enzymes. But then again, it also poses a problem, whereas it has to be removed when the virus enters a plant cell before the virus can spread. The capsid protein.
Now we will discuss about the structure of DNA. As we know, DNA is the deoxyribose nucleic acid. The DNA helix has uniform width of 2 nanometers and adjacent bases are 0.34 nanometers apart. Most DNA double helices are right-handed. That is, if you were to hold your right hand out with your thumb pointed up and your fingers curled around your thumb would represent the axis of the helix and your fingers would represent the sugar phosphate backbone. Only one type of DNA, which is the Z-DNA, is left-handed.
Hydrogen bond between the two bases pairs make the DNA molecule much more flexible. Hydrogen bonds are not covalent bonds, so the DNA molecule can break and form relatively easily. For this reason, DNA can be separated like a zipper, either by a mechanical force or high temperature. So, small amounts of energy is required to break the hydrogen bonds between the base pairs. DNA also stabilized by effect of hydrophobic and stacking. Therefore, the internal and external effect of hydrogen bonding are important to stabilize the double helix strands. A polynucleotide is simply a long chain of nucleotides. Poly means many, so polynucleotide literally means many nucleotides. DNA nucleotides are linked by having a phosphate shared between two deoxyribose sugars on adjacent nucleotides.
Phosphate is a very simple molecule made up of a phosphorus atom bonded to 4 oxygen atoms. This molecule is reactive, meaning that it wants to be bonded to another molecule. Deoxyribose is a sugar that has 3 hydroxyl groups OH that are also reactive. These OH groups are important because it is here that the phosphodiester bonds form, linking each deoxyribose to the phosphorus. There are 2 ester linkage, so each deoxyribose has 2 available OH groups and can bind to 2-phosphate, creating a chain, aka the backbone of the DNA.
When it comes to the TMV infection, the protein coat is very helpful in that it protects the RNA from being destroyed by enzymes. But then again, it also poses a problem, whereas it has to be removed when the virus enters a plant cell before the virus can spread. The capsid protein has several clusters of acidic amino acids, which are stable outside of cells where the calcium levels are high, but inside the cells where the calcium levels are low will repel each other. So this repulsion will loosen the first few capsid proteins, hence releasing the end of the RNA. The ribosome of the host plant.
This structure places the non-polar bases of DNA in the center of the double-stranded molecule, surrounded by the charged phosphate groups. First, remember that like charges repel each other. The double helix structure, with negatively charged phosphates on the outside edges, allows the phosphates to be as far apart as possible. Second, the non-polar, uncharged bases are hidden in the center of the helix. The cellular environment is aqueous and therefore polar, so surrounding the non-polar bases with charged phosphates maximizes the solubility of DNA under physiological conditions.